Hello everyone, good afternoon. Welcome to this online classes from Ahmadiyya International School, and this is Zafar, and we are going to continue with our class of protein synthesis. This is part 2, where we will be learning about translation. In part 1 we discussed about transcription. Now we will discuss about translation, the process that takes place in the cytoplasm. Before we go into translation part, let us discuss a few important things about our genetic code. Our genetic code, it has some characteristics, or you can say the features. We are going to discuss some features of our genetic code, how the genetic code works and what are the rules first of all. First thing you have to remember, our genetic code is a triplet code. Our code is degenerate or redundant. Our code is universal. The genetic code, so please remember when I say the genetic code, the way the nitrogenous bases work actually, and then our code is non-overlapping. So these are the 4 important features. So our genetic code is triplet, and it's a degenerate, and it's universal, and it's non-overlapping. Let us go into the details of each one of these. So first thing, a triplet code. What do you mean by a triplet code? A triplet code means 3 nitrogen bases on mRNA or on the DNA code for an amino acid. So if I show a piece of mRNA here, it will be coding for 1, 2, 3, 4, 5 amino acids, not more than that. So you might ask a question, why is our code a triplet code? So I have to explain you with a very simple example here. So before I go to the example, let me tell you, if the code were singlet or a doublet, what will be the drawbacks of such a code? So why is not our code a singlet code, or why is not our code a doublet code? Remember one thing, the nitrogen bases that we had in the DNA were only 4 nitrogen bases, that is adenine, thymine, guanine, and the cytosine, the nucleotides containing the nitrogenous bases. But we already learned how many types of amino acids are there. We have 20 different amino acids. So you can see there is a mismatch between the nitrogen bases and the number of amino acids. If they were same, for example, if there were 4 nitrogen bases and there were only 4 amino acids, the problem would be solved. There would be only a singlet code. Singlet code means each nitrogen base coding for 1 amino acid. So to make you understand in a much better way, I'll take one example here. Let's take one example where there are 20 students sitting in a class, and I have 4 different markers, and I have to call them to the stage. There are 20 students. I have to call these 20 students to the stage without using their names, without using any language. I have to call them to the stage. So generally, normally if I have to call some student, I would say Amade come here, or maybe Ibbe come here, or Aisha to come here. I'll use their names. But in this case, I'm not going to use names. I have to use a code. So let's say I'm using markers, 4 different colors. So if I use one color, one student comes to the stage. If I use another color, another student comes to the stage. If I'm using 4 colors, I'll be able to call only 4 students to the stage because I have no other color. But there are how many students there? 20. So I will not be able to call 16 students, they will be left out. So I can't use one color code for each student. So a singlet code is not possible. Now you might say, why not use? So though I have 4 colors, but I have many of each color. So I have black so many, I have green so many, I have blue so many, red so many, but only 4 colors. So if I'm using, let's say, combination of colors, but I'm going to use only 2 combinations, that will be called as a doublet code. So if I'm using a doublet code, that means 2 colors, red and red one student, green and green another student, green and black another student coming to the stage. So if I am using 4 different colors and arranging them in different combinations, you know the combinations that I get is only 16 combinations. black and black, green and green like this, I get only 16 combinations. There will be 4 students who will be left. I will never be able to call them to the stage because I don't get enough combinations. The next possible option or the best option is to use a triplet code. A triplet code means using 3 colors to identify each student. So here in this case, I am using 3 different colors just to identify or call one student to the stage. So black, 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 one student, black, black, green, one student, green, green, black, another student coming to the stage. So in that case, if I am using 3 to call one student, I will be able to get 4 times 4 times 4, I get 64 combinations, arrangements of 4 different colors. So I get 64 combinations, but I have only 20 amino acids. So this solves the problem. That's why our genetic code is a triplet code, 3 nitrogen bases coding for 1 amino acid. Okay, so you have a triplet code, not a singlet code, not a doublet code. So how are you going to write it in the exam? In the exam you are going to write, if you are using a doublet code, the combinations that you get of doublets is only 16, but there are 20 amino acids. Why not a singlet code? Again the same thing, there are only 4 nitrogen bases, but there are 20 amino acids. So there is a mismatch. The best possible option is to have a triplet code where you have 3 nitrogen bases coding for 1 amino acid. So there are 20 amino acids, you get 64 combinations. So you have more than required combinations for if I use a triplet code. The next thing that we are going to discuss is the degenerate code. As you have seen, we have 64 combinations possible with a triplet code, 64 combinations, more than required actually. So these excess combinations, what am I going to do with these excess combinations? So I have allocated for 20 amino acids, there are remaining 44 codons, or you can say the triplet codes which are excess. Do you agree? 20 given for 20 codons given for 20 amino acids. I am left out with more 44 more. So should I just ignore them? Just ignore them or just leave them like that? No, it's not possible. I have to use these 44 codons also. So out of these 44 codons, remaining codons, after 20 been given to 20 amino acids, 40 again are used for the amino acids only. 40 again used for amino acids. 4 have a special role to play. We'll discuss that special role. There will be the stop and the start codons. They signal the process of starting and stopping the translation process. We'll come to that special role. But this 40 plus this 20, that means 60 codons out of 64 codons are used for amino acids. That means each amino acid, for example, leucine has about 6 codons. More than required, it's not only 1 codon. So you might say, sir, what is the definition of a degenerate code, or how do you explain this? Degenerate code means each amino acid has more than 1 codon. To explain you in another simple way to understand this is, let's say I am given 64 names. Names to be given to newborn babies. I went to the hospital, there are only about 20 infants there. So, but I am having 64 names. So what am I going to do with the 64 names is, I may give 20 names to each infant, I am still left out with 44 names. So what I do is, I give each baby one more name. Let's say Ibrahim or Ibbe, Ahmad or Amade. So I give two names. Even if I call Ahmad or Amade, the same guy comes to the, I'll be referring to the same guy. Same thing, each amino acid is given more than one name, or you can say more than one codon, not name, sorry. Each amino acid is given more than one codon, so that they are called for translation. So again I repeat, degenerate code is each amino acid has more than one codon. So you might say, what's the advantage of this? So can you explain a little bit better? For example, CCC codes for proline, CCA also codes for proline. Proline is an amino acid. CCU also codes for proline, as well as CCG also codes for proline. Let's say it be because of a mutation, even if the last nitrogen basis is mutated or it has a different sequence, still it will code for the same amino acid. It doesn't matter what you place at the last nitrogen base. I think you got it. So degenerate code helps to protect you from the harmful effects of mutation, especially at the last nitrogen base. So that's called a degenerate code, which means each amino acid has more than one codon. The next thing is, the next feature of our genetic code is universal. Universal means all the organisms, starting from a bacteria up to human beings, they all follow the same genetic code. A mosquito or a plant or a banana plant or a coconut plant, you name it, all the organisms on the planet, they all follow the same rule, the same set of rules. So let's say when I say CCC codes for proline in human beings, CCC codes for proline in all the organisms, it's universal, more or less universal. So that means in theory, it's very interesting here, in theory I can take a gene from one organism and place it into another organism. So I can make transgenic animals. If I look at it, let's talk about one very interesting example where scientists took the gene from a firefly, which helps to make the fire. The gene was taken out from a firefly, which is an insect, and they inserted the gene into a plant. Which plant? They inserted into a tobacco plant. Guess what happened? In this case, what happened was, this firefly gene was expressed in the tobacco plant, and you had this tobacco plant or the tobacco plant glowing in the dark. It's very interesting that you have a plant which is glowing in the dark. So you might say it's very interesting, so we can keep these plants just in front of our house and we can see them glowing in the dark. But actually scientists use this firefly gene for another purpose actually. So they wanted to study the gene expression. Wherever you see the tobacco plants which are having the tobacco plant parts which are having that glow in the dark, that means the gene is getting expressed there. So they wanted to know about which parts of the tobacco plant are expressing the genes during different stages of development. So it was a very good example of showing that the code is universal. Plants, animals all share the same code. And I think this investigation was done in 1860, I guess, long back. Not 1860, I'm not sure. You know very well how good I am with numbers. Once I was explaining this William Withering experiments, his drug trials. In one of the classes, there was a boy who was very good at mathematics. Very good, I would say. He never uses the calculator also. He was very good at that. So once I was explaining this William Withering experiment, and I told him that he did his experiments in 1875, something like that, in the class. And I told maybe plus or minus 1 year, just like that. So this guy, the mathematics guy, he's so good at maths, and he always thinks about numbers. He never uses a calculator also, mentally doing the numbers. And he was looking at me like this, okay, so let me see like that. The next day when I was explaining, when I came, I was explaining about something else. This guy was shaking his head, like anything. When I asked him, why are you shaking your head? This guy said, sir, you said this William Withering did his experiments in 1875. I said, yeah, maybe around 1 or 2 years, plus or minus. That guy said, sir, you missed the year by 100 years. This guy did his experiments in 1775 or something. Then I said, my goodness, this bioteachers, we can't remember the years basically. So here I'm not sure about the year here. 1960 or maybe around 1860, I think 1960 he did his experiments, and these are very interesting experiments where they took, I think it's 1960 only. So they took this gene from a firefly and they put it into a plant, and the gene was expressed in the plant itself, and that is possible when the code is universal. So plants, animals, microbes all follow the same genetic code, the triplet code, and it's a degenerate code and all. The next thing that we are going to discuss is our code is non-overlapping. So what is non-overlapping? So I can take an example for this. So if I take non-overlapping code, if I take a sequence like this on the mRNA, what do you expect? How many amino acids will be coded by this sequence? If it's a triplet code, you know very well. You might say, sir, there are only 2 codons in this. The first codon is CGU, that codes for 1 amino acid. The next codon being CAC, this codes for second amino acid. So this, basically this will code for 2 amino acids. What if I tell you the same code, if it were overlapping, it would be coding for 4 amino acids. Let's say CGU, the same code, CAC. Let's say it's read like this. The first codon is CGU, first codon. Second codon is GUC. Third codon is UCA. And the fourth codon is CAC. So the code is read like this: the first codon, the second codon, the third codon, and the fourth codon. So if the code were like this, it would be called as an overlapping code. If the code is like this is one codon, this is another codon, this is called as non-overlapping code. What do you think is the code that our cells follow? Our cells follow non-overlapping code. It's not overlapping. Are there any organisms which have overlapping code? Yeah, there are some genes in bacterial cells which are overlapping, and there are some organisms as well, but only few genes are overlapping. But majority of our eukaryotic cells, they have a non-overlapping code. So how do you define a non-overlapping code? Is 3 nucleotides or the nitrogen bases, they code for 1 amino acid. The next amino acid will be coded by the next 3 nitrogen bases. That means this nitrogen base or any of these nitrogen bases do not become a part of the next codon. CGU is different from CAC. But in this case, the nitrogen bases of the previous codon, these 2 will be used in the next codon, and these 2, the last 2, are used in the next codon. So this kind of code is called as an overlapping code. Now you might say, which one is more advantageous, which one is good? If you look at overlapping code, short sequences of DNA or RNA can code for many amino acids. You see, short sequence, 4 amino acids coded. Here it's lengthier, but still it coded for only for 2 amino acids. So this will be the advantage, being short sequences can code for many amino acids, but there is a serious drawback with this. The serious drawback being, if I take this sequence, I will be restricted by which codon will follow after a particular codon. That means the proteins will have less variety in that. I think you got the point. I will be restricted by my next codon because I am going to take 2 nitrogen bases from the previous codon. So I can't start this codon with CCC because I am going to take GU from the previous codon. So this is a restricted code, or you can say the proteins will have less variety. Let me just recap this one. What are the characteristics of our genetic code? Our genetic code is a triplet code. 3 nitrogen bases code for 1 amino acid. The next thing, our code is degenerate. There is more than 1 codon for a particular amino acid. Next thing we learned, the code is universal. Universal means all the organisms on this planet, they follow the same code. Last one, our code is non-overlapping. That means nitrogen bases of one codon are not used in the next codon. They don't become a part of the next codon. So these are the characteristic features of our genetic code. Okay, now let us discuss about translation. Now, after finishing about the genetic code, let's go to translation part. What is translation? Remember, translation is first of all there will be transcription, then you have the translation. Here is where the mRNA is made, that we discussed in the previous class. Now in this class we will discuss how amino acids get joined to one another to form a protein. And this happens in the cytoplasm. Remember the translation. Before we go into the details of translation, let us discuss about the structure of some important molecules. Like, we'll discuss about ribosomes, which are organelles. In the cytoplasm, they help in protein synthesis. We'll discuss about tRNAs, very, very important structure of these two. Now, if I talk about ribosomes, ribosomes are made up of rRNA and proteins. Chemically, they are made up of RNA again nucleic acids plus proteins. They have two subunits. One is called the smaller subunit, the other one is called as the larger subunit. Two subunits which are present separate, but once the mRNA comes, they attach and join together. But otherwise, they will be separate entities lying in the cytoplasm. They have sites for binding of mRNA. So, they have mRNA binding sites where the mRNA will attach here. Then they have binding sites for tRNA. Okay, so these are called as EPA sites. No need of remembering that EPA sites. So here you have spaces for the attachment of tRNAs. Okay, at any given one time, remember, two tRNAs will be present inside the ribosome during translation. So this is the ribosome, this is the larger subunit, and then you have the smaller subunit. Together they form the ribosome, and it has binding sites for mRNA, it has binding sites for two tRNAs. So they have sites like EPA sites are there. No need to no need to remember that EPA sites and all. You can just say they have binding sites for tRNA as well. And we have eukaryotic cells have ATS ribosomes, whereas prokaryotic cells have 70S ribosomes. That will be enough for the ribosomes part. So if they ask you what's the function of ribosomes, you can say ribosomes are the places where the mRNA binds and then amino acids are attached to one another. So this is the place which will bind the tRNAs and the mRNAs together, and these are the sites of translation. So where do the translation takes place? On the ribosomes. Either the ribosomes are freely present in the cytoplasm, or you know very well they'll be attached to the endoplasmic reticulum. That will be called as the rough endoplasmic reticulum. The next thing is about tRNA. So tRNA, how does it look like? So this is a tRNA molecule. I am drawing very roughly here. Okay. So here you have, actually it's a single strand of RNA which has been folded like this. Okay. So don't say it's a double helix or anything. So it is folded, it has loops, okay, and it has binding sites for amino acid. Amino acid binding site on one end. On the other end you have 3 nitrogen bases hanging out like this, or projecting or protruding out. For example, here CGU. This is called as anticodon. Okay, it will be complementary to the mRNA codon. We'll see that later. So here you have, what are here means, here you have complementary base pairing taking place like this. So these are the hydrogen bonds, and these are the nuclear. This is the sugar phosphate backbone again. So here you have cytosine guanine, uracil adenine, so double bond, triple bond, the same thing. So basically it's a single strand of RNA molecule which has folded into a tRNA molecule. tRNA meaning transfer RNA. Why is it called RNA? It's a nucleic acid which is made up of nucleotides. It's also made up of nucleotides. But it has special sites here. One is amino acid binding site, it has anticodon, it has hydrogen bonds which are lacking in mRNA. So what is the role played by tRNA? As the name suggests, the transfer RNA, what does it do? It transfers the amino acids from the cytoplasm to the site of protein synthesis, which is the ribosome. So they transfer the amino acids. From where to where? From the cytoplasm to the ribosomes. They release them there, again go back, fetch the same type of amino acid. And remember, there are some very, very important points you have to remember. How many types of tRNAs are there? So you should say there should be at least 40 to 60 different types of tRNAs. Remember that. There are more than this. I mean, around 40 to 60 different types of tRNAs are there. You might ask, so why those many tRNAs are there? Remember, our code is degenerate. There is more than one codon for an amino acid. That means for each amino acid, there might be more than one tRNA that will transfer it. I repeat again, we have more tRNAs because we have more codons, more than required. Remember, we had 64 codons there instead of 20. So that excess codons, that means we should have excess tRNAs. So each tRNA carries only one type of amino acid. That's why we have so many different types of tRNAs. Okay, so remember the point. One amino acid can have many codons, but one codon codes for only one amino acid. Please remember, this is very, very important. So each amino acid, for example, there might be 6 tRNAs that can carry leucine, okay, but each codon, let's say AUG, always codes for methionine. It's for only one amino acid. This is unique. Okay, that part you have to understand. Let's say they ask you differentiate between mRNA and tRNA. That's a very important question. If you differentiate between mRNA and tRNA, or if they ask you to compare, both are, compare means you can write the similarities as well. Both are ribose nucleotides, you remember, they are ribose nucleic acids. And if you are writing about the differences, it's very, very important. The differences being, remember, mRNA is a straight chain compound, this is folded. mRNA can have variable length. What do you mean variable length? It depends, the mRNA length depends on the length of the gene, by the way. If the gene is lengthy, mRNA will also be lengthy. tRNA, they have a fixed shape or a fixed length, they don't have a variable length. Do mRNA have hydrogen bonds? No hydrogen bonds. What about tRNA? They do have hydrogen bonds. And a very important thing, they don't have any no amino acid binding sites. They don't have any amino acid binding sites, but tRNA, they have amino acid binding sites. Last one, they carry codons, they have anticodons. This part is very, very important. Differentiate between mRNA and tRNA. This is a straight chain compound, this might be folded, this may have variable length depending on the gene length, and this will be fixed length. No hydrogen bonds there, they do have hydrogen bonds. No amino acid binding site, but they have amino acid binding sites. No, they have codons and they have anticodons. Okay, so the structure also is very important for tRNA. Remember, amino acid binding site is there, anticodon is there, they have loops, they have hydrogen bonds here. So they have a clover leaf shape. Now let's directly go to translation. Okay, now let's go to the translation part now. So as you can see here, this question comes for 5 marks generally. Describe the process of translation for 5 marks. So how are you going to write this? As soon as the mRNA, this is the mRNA molecule, which is a copy of the information on the DNA, as soon as it comes into the cytoplasm, the ribosomal subunits, the smaller and the larger subunits, they bind with the mRNA. The smaller and the larger subunits, they bind and they engulf 2 codons at a time. Remember, there are always 2 codons within the ribosome. And the beginning of the mRNA, you have a start codon, which is always AUG. Remember, AUG on mRNA is the start codon. What it means is, it tells or signals the ribosomes to start the process of translation from this point onwards. It doesn't code for any amino acid. Then you have, at the end of this mRNA, you have a stop codon. So here you have 3 stop signals, either of them, any of them. It can be UAA, UGA or UAG. These are stop codons, they will be present at the end of the mRNA, this is at the beginning of the mRNA. And here you have, this is one codon, one triplet code, coding for one amino acid, second triplet code, third triplet code, like this. So this code will be coding for 1, 2, 3, 4, 5, 6, 7 amino acids. Okay? So what happens during translation? First of all, I told you mRNA comes and the ribosomes bind with it. Each tRNA attached to a specific amino acid. What I mean is, for example, this is not correct basically. Here, if you take about this anticodon GCG, it will be always connected to a particular amino acid. Okay, in this case, it's histidine, for example. So this codon, this anticodon always binds with histidine only. That means this tRNA is given the task of bringing only histidine. So they bring the amino acids from the cytoplasm to the ribosomes, leave the amino acids here, go back, fetch the same amino acid again wherever the same complementary base is there. So these tRNAs come and bind, they brought the amino acid with them. They bind inside the ribosomal subunits, and another tRNA, now you can see this, they brought the amino acids here. When two amino acids are lying side by side, you know the amine group of one amino acid can form peptide bond with the carboxylic group of the next amino acid. So these two will form a bond, name the bond, you get a mark for writing this. So they form a peptide bond by condensation reaction, and this tRNA leaves the ribosome. Now the point is, will it carry the amino acid or will it leave the amino acid there? It will leave the amino acid there itself, and this one leaves, gets out of the ribosome. The ribosome moves one codon at a time. So the next place, the ribosome will be here, the subunits will be here, and this tRNA binds into this space. Again remember, you have to write this as a marking point. Complementary base pairing rule takes place, or you can say complementary base pairing takes place between codon on the mRNA, anticodon on the tRNA. So they form bonds here again, but remember these are temporary bonds which can be broken again. Again this tRNA leaves, the ribosome moves until the end of the mRNA is reached and the ribosomes dissociate, and you got this sequence of amino acids connected like this. So if you ask me about the summary of this, look at this. You have the DNA. Using this DNA, the cell makes an mRNA, and using this sequence on the mRNA, amino acids get attached to one another. And there is a specific way for, it's not like randomly they are getting attached. They follow certain rules, and then you get the protein molecule, and this is called as central dogma of protein synthesis, or central dogma of life, where DNA gives rise to mRNA. This mRNA is used to attach the amino acids, and ultimately you get the protein. So which amino acids get attached depends on what type of sequences are there on the mRNA, and what sequences are there on the mRNA, they depend on DNA. So this is about translation. I hope this will be understood, but if you have any questions, I already told you, you can message me on Viber and we can discuss it in the panel group. Thank you very much.